I would like to welcome everyone to this webinar about the Decode ME study. We are very happy to be joined by Professor Chris Ponting from the University of Edinburgh. And with me, I also have Margrethe Gustafsson, our communications advisor, who will help me keep track of questions and just keep me on track as well. You know, ME can be difficult at times. So just a little bit of housekeeping. The webinar will be recorded. It will be posted to our website and to YouTube and Facebook afterwards. You can ask questions. Please use the Q&A function at the bottom of your screen. We will look through the questions. If there are many questions about the same thing, we'll try to group them together to make it more coherent. So then I just want to say welcome to you, Chris. Very happy to have you here. This study has created a lot of interest and quite a few articles in the Norwegian press as well, which is quite uncommon when it comes to ME. And but before we start, just to get a little bit of your background, how did you get interested in the ME field?
To start off with, thank you very much for the invitation to tell you all about the Decode ME results today. I got interested in ME because my good friend Simon, who I got to know in university in the mid-1980s, his life has been devastated by ME. His hopes, his future, his expectations of life were completely destroyed by the disease. And I started off in my academic career as a physicist, never thinking that I would be able to do anything on ME, but gradually as I got closer to biomedicine, I thought I might help. And when I entered the first meeting of ME, I was shocked by how few people there were there. Shocked by how few studies were being done, particularly those that were large in scale and power. So I thought I would try and help, and ultimately this ended up in this project, Decode ME, which is very much a co-production with people with ME. So it's been a long journey, but really it comes from some understanding, personal understanding, but it's in the end, it's been a privilege to work on this disease.
Well, we are always grateful for any researchers who take us seriously. So thank you for doing this. You have a presentation for us as well.
So I hope you can see my slides.
Yes.
And I'm going to hide those controls. Right. So yes, it's been a real privilege to be part of this study, and thank you very much for the invitation. So Decode ME has been running in its funding period for almost 5 years. It's been a co-production with people with ME, the charities led out of Forward ME by Action for ME, and the whole study has been funded to the tune of about £3.5 million by the UK Research Council called the Medical Research Council and the National Institute for Health and Care Research. So I'm going to tell you something about why we thought that genetics was the first thing that should be done. A little bit about the journey from there to here, then the results, which, as you know, came out in preprint form. So they're initial results, and they came out at the beginning of August. I'm going to say a little bit about what they mean and perhaps what they don't mean, and I'm very happy to discuss any aspect in the Q&A afterwards. A little bit after that about what's next for the project, because as I said, we haven't finished. And then what's next for the DNA samples and the data, and then we'll go into the Q&A session. So to start off with, why is it that genetics might be important to understanding ME? One major reason is that for diseases generally, it has been shown that when there is genetic support for a drug target, this doubles the success rate in clinical development of that drug. So in other words, the pharmaceutical companies nowadays require genetics to support their funding of, which is very considerable, for their funding of drug development. And so we knew that we needed genetics to support future clinical development of drugs. But a second and very important reason is that the type of genetics we do is unbiased, and it's comprehensive in the sense that we look everywhere across the whole of the genome. And when I started reading the ME literature, I think it's fair to say there are huge numbers of hypotheses, but not often is it that the results from one study is replicated by another. So in that circumstance, I think it's really important to step back and say, let's put all of those hypotheses to one side and just ask the question, among all of the genes across the whole of the genome, what are the genetic risk factors for ME? So these are answers that are not rooted in what we already know. And so I think that we know in biology relatively little. And so we can begin to find things and aspects of biology that perhaps are completely unknown today. And then finally, what genetics can do is to indicate what bodily system is most involved with respect to genetic cause. And you all know that the symptoms of ME are very diverse and they can fluctuate over time. And so this is quite important to try and work out where exactly in the body do the genetic causes reside? Where do they come from? Because ultimately we need drugs that are targeted to the root cause and not downstream consequences. So we also knew that researchers had found that multiple members of the same family can be diagnosed with ME, but that these diagnoses do not follow a predictable pattern. And if they had followed a predictable pattern, then it would have been a Mendelian disease, and we would have expected one or a couple or whatever of genes that were disrupted in ME. And because it's not like that, it is a so-called complex genetic disease. And complex genetics are all about small effects on many genes. So right from the beginning, we thought, and this turned out to be true, that we wouldn't find a single gene, but we hoped that we would find many associations pointing at many genes. And in our study, the Decode ME study, we measured something which is called heritability. It doesn't really matter what that is. But if that thing that we measured was 0, and it is not, if it were to have been 0, then there would have been no genetic contribution to ME. But that wasn't the case. So it doesn't really matter what the number was that we found, but it does matter that the estimation of this quantity, heritability, is not zero. It even thinking about its error of 0.6, this 9.5% value means that we have demonstrated that genetics do contribute to ME risk. So at the heart of our project, Decode ME, and hopefully a future one, Sequence ME, is public and patient involvement. This has always been the case. Indeed, the first meeting of ME that I referred to earlier was a meeting between researchers and people with lived experience of ME. So it's a very natural thing for me anyway to think about, but quite an unusual thing to do, is to work alongside people with lived experience. And I cannot say enough really about how much PPI improved the science. It improved the science because I learned a lot as a researcher and hopefully fed that back into the project. It improved because with people with lived experience, we could gain the trust of our participants and the large number of them that were required. Not because we had PPI, but because we were able to say things that resonated with people with lived experience, who then realized that we had an understanding as a project, even if I at the beginning did not. We had an understanding as a project what it means to live with ME yourself or someone close to you. So here are some circles, and each circle is a team, and each team has someone with lived or more people with lived experience. In the middle is the steering group, the PPI steering group, and the top right are the management team, of whom I'm just one, and the other two have lived experience. So there was one famous occasion when a decision was taken by the management team, and I was outvoted 2-1. But that's, in a large way, something I'm proud of, because, I mean, I can't remember what it was. I'm sure I was wrong, but it allowed the lived experience to pervade everything that we do, right up to making major decisions. Now I'm going to show you this rather complicated slide, and I'm going to go through it rather slowly and apologize that it is complex. It's taken from this publication that's mentioned in the top right. So you'll see at the bottom that what it is a set of graphs over various years. And on the left, in blue and green and in brown, are different things. And so there are three y-axes here. And I'm going to start on the left. These are the number of publications in blue of genome-wide association studies over time per year. So in blue, we have, starting off with something like 100 or so publications on genome-wide association study 18 years ago, 2007, peaking here and 400 and coming up to 600 or so in 2021. So in other words, this time from 2007 onwards was the time when genome-wide association studies were being applied to all of these different diseases, and hundreds of different publications were being done, with one obvious exception. There were no large genome-wide association studies for ME. So in my view, ME complex genetics has lagged 10 to 15 years behind other diseases. It should have been done 10 to 15 years ago, and ME should have gained the benefit of those results 10 to 15 years back. And we're running now to catch up with all of these other diseases, which are making headway and breaking through and are helping to find effective therapies for other diseases, but not ME. The next one is the average number of signals in green. And so here is about 10, and here is about 20. And so, in other words, at the beginning, these studies were finding about 5, and then 10-ish, and then 20 or so, and up to 30 and 40 in more recent times. And the reason for this is the average number of samples, starting with quite a low number, which is in brown, and then taking off really about 10 years ago above 20,000 individuals. And this is important because the larger the study, the greater the power. With the greater the power, the more associations, the more signals are found, and the more likely it is that there is a genetic signal found that can have an intervention quickly. So a drug can be found from the genetics more quickly with larger numbers of signals. And then lastly, you will know that we reported 15,500 samples from European individuals. We have 18,000 DNA samples, including other ancestries. So say 18,000. So therefore we expect roughly around 5 discoveries, 5 signals with that number of samples. So in my view, Decode ME is just the beginning, and we need other genome-wide association studies, both to increase the power overall, because you can put the data together, but also to replicate the findings that Decode ME has found. Okay. So what is this genetic study that I've been talking about, a genome-wide association study? In essence, it's quite simple. It's asking at a single point in our genome, the human genome, where there is either a letter here, C, or a T. So there are 2 different variations at this place on our genome. And when you separate those people who are cases, here it's heart disease, and you ask how many of those people have a C and how many of those people have a T, and then separately you do that for other people without that disease, and you ask how many people have C and how many people have T. And then it's, after that, statistics, asking whether this change, that there are more people with C than T than seen in the controls, is that significant? Is that statistically significant? And even though it may be significant when you test one place, you have to think about the fact that you're testing over hundreds of thousands, if not millions of places. And so you'll get lots of false associations merely by chance. So to overcome this rather heavy burden of statistical power, you have to use very large numbers. And for the genome-wide association studies that we're talking about today, it's just over 15,500 cases. And we used for our controls over 250,000 people with European ancestry who had volunteered for the UK Biobank project. And so we didn't need to spend any money generating the genetics of the controls, because we could simply use the available data for doing so. So we focused our money on the cases. And here's an example, not of ME, but of a different disease, just to orientate you as to this plot, which is called a Manhattan plot. And it's called a Manhattan plot because where there are signals, these look like skyscrapers. So you can see here that there are 3. There's a huge one on chromosome 11, and then a couple of smaller ones on chromosome 6 and 4. So the x-axis is walking along each of the chromosomes in turn, and they've been colored alternately. And on the y-axis is a measure of the significance of this statistical test. And there's a negative log 10, but essentially the more significant the finding, the higher the skyscraper. And these three signals burst through a horizontal line here, which in the industry of population genetics is a very sound, very well understood threshold that everyone says that anything above that is genome-wide significant and should be believed. Anything below that isn't necessarily not true, but neither is it demonstrated to be true. So in this study on Lyme disease, there were three associations. And so this study asked the question, did we expect these? Well, they did indeed expect an association in this part of chromosome 6, because this part of chromosome 6 is well understood to affect people's susceptibility to infectious and autoimmune disease. This part of chromosome 4 was expected because changes to a gene, TLR1, had already been found to be associated with Lyme disease. But the big peak had not been expected and was found to be a secreted molecule that affects the ability of the bacterium Borrelia burgdorferi and affect its growth. So that immediately gave an explanation for why some people are more susceptible than others, because some people have more effective control of the bacterium than others. Right. So I've told you about how Decode ME started, and it started essentially 10 years ago through trying to get the funders interested, writing down what we thought of as being needed, receiving questions back saying, you know, we don't fully understand or appreciate or believe you, and then answering those questions, persevering, in other words. And we were helped to a great degree by many people because it was the case that not all of the doors were open for us to go and speak to the right people, and several people opened the right doors for us. We put together a funding application with 3 objectives, and this was ultimately the funded project: to identify genetic differences in people with ME. And these would begin to explain biological mechanisms. We never said, and we don't believe, that this is the end of the journey. It's the end of the beginning of the journey. Secondly, we wanted to build a cohort of people with ME so that other researchers could make use of that cohort. And that is the case. We do have that cohort. They are available for researchers. And then thirdly, we wanted to disseminate the data where we have consent to academics and to industry, and we are doing that, and will continue to do that. I've explained how we've co-produced the project, and we recruited from the beginning 27,000 people above the age of 16 with a clinical diagnosis of ME who live in the UK with post-exertion malaise, either CCC or IOM criteria. These criteria were chosen by people with lived experience, and the ways in which post-exertion malaise was defined drew upon people's lived experience. So the cohort came from people who were interested in the study to begin with. 27,000 people completed the questionnaire. 22,000 or so met the eligibility and were sent collection kits. 18,000 returned the saliva collection kit. Now, by the way, that's enormous. A success rate of 18,000 among 21,500 just demonstrates the enthusiasm of people with ME for participating in research. DNA was successfully extracted and passed quality control for 16,700. And for this initial analysis, 15,500 of those DNA samples were analyzed because they have a single ancestry, which is in this case European ancestry. We are continuing to do research on the whole cohort, irregardless of ancestry. The age range of our participants roughly relates to the people in England with a G93.3 post-viral fatigue syndrome diagnosis, and roughly agree with what people in that English cohort say with respect to their comorbidities, with one exception, which is irritable bowel syndrome. We don't know why people with Decode ME in that cohort have twice the likelihood of IBS than the English people. We published on the symptoms, so I'm not going to dwell on this. Obviously, those people who participate in the DNA part of the project all had post-exertional malaise. So the further you get from the middle of these circles, the more a particular symptom was told us by the participant. So people with pain, noise susceptibility, gut symptoms, brain fog, confusion, unrefreshing sleep, these are, and fatigue obviously, are all symptoms that you will recognize as being highly prevalent among people with ME. So this is the Manhattan plot for the initial Decode ME analysis. So we have announced 8 genetic signals, and they are indicated by using a single gene label, but there are many genes under each of these peaks. So 6 of them were found in our primary GWAS, and 2 of them, which are these 2 here in chromosomes 12 and 13, were found in subsequent GWAS. But you can see there is a strong signal for each of those. They just don't break through this genome-wide significant threshold. So we have 6 that came through in our major GWAS. The other 2, the 12 and 13, were found in GWAS 2, and one of those in something called GWAS infection. I'll take you through these right now. So our primary GWAS involved everyone. Our GWAS 2 is a GWAS that is in preparation for us trying to replicate. And there's a technical reason why we needed to do the second one, and that was all about holding to one side our controls, or subset of our controls, so that we could do the replication. But I won't settle on that and talk about it further because it's technically difficult. Now, only those people who said they were female, sex at birth, of course, they were 85% or so of our cohort. Therefore, the statistical power is very strong. We saw 3 of the 6 signals for the female GWAS. We found none of these signals in GWAS male. For those people who reported an infection at onset, we found 3 signals, including one of the 2 that were only found in GWAS 2. And I'll come back to this one because this one is very strong in the GWAS infection and is near to a gene that may yet explain why. When we looked at those people who did not report an infection just prior to onset, we did not find any of these associations. So looking at the 8 associations, there are some broad results and conclusions we came to. First of all, that the genes that lie under these signals indicate the involvement of two different bodily systems, the immune system and the nervous system. Two of the signals, at least, relate to the body's response to infection. I'll just point you to the BTN2A2 on chromosome 6, where it and several surrounding genes are part of the sensing process by which humans and other animals detect bacteria. And then the OLFM4, which I just referred to previously on the GWAS infection, this one, which is significant in that GWAS, lies next to the OLFM4 gene, which in neutrophils, its role is to dampen down the immune response to infection. And so if it doesn't dampen it down, then the immune system may run very much in a dysregulated way. Other signals point to the nervous system. I'll come back to that. And one had been previously found among people experiencing chronic pain, and that is the CA10 one on chromosome 17. And I'll show you that in a minute. But overall, looking at the genes, looking at our interpretation of the results, we were, I think, able to say with strong evidence that the signals align with how people with ME have described their illness. With respect to symptoms after infection, in large part, with respect to nervous system involvement, and this coincidence of people's genetics to do with chronic pain and the genetics of ME, which I'll come back to. When we ask the question, are these genetic signals different between males and females, the answer was we had no statistical evidence for that. So at the moment, there's no evidence for female-based genetic risk factors being any different from male ones. The caveat is that we have not yet investigated the X chromosome, and that is ongoing, but we didn't have time to do that analysis and to do it well enough for the preprint. And then lastly, we have these 8 signals, and we had colleagues elsewhere in the world to look in their data to see whether they could also see these genetic signals. And for half of them, there was some evidence, but it's modest, some evidence of replication in their data. So we didn't strongly replicate these signals, and in part that could be because there are very substantial differences how we brought together our cohorts and how these different cohorts were brought together. Now, I'm going to say something about, in genetics, something called co-localization. And essentially what this is trying to answer the question, are two genetic signals overlapping? So they trace the same curve. So as you walk down a chromosome here, then what you see is the same peak as you would for one trait or disease over another. And that might imply that there is the same causal genetics causing both traits, that is one gene, say ME genetics, and something else, let's say pain. So on the other hand, we may see two peaks which are quite close to one another, but they don't trace the same path. And so they are different from one another, and so they have different genetics. And we have to do this because many of the DNA differences that we carry around with us are co-inherited. That means that they go down the generations together rather than being separated. And that means that there are large chunks of our genomes that we cannot separate out the genetic signals. And so this relates to this image of a figure in the preprint, which is trying to show that in across the different chromosomes, what we see in green, which is the Decode ME genetic signal, does not overlay the genetic signals here for chronic pain in chromosome 1, or depression, chromosome 1, chromosome 13, clinical depression, 20, depression. But when we look at chromosome 17, there is a peak for chronic pain. And when we look very carefully, they do overlay one another. And so there is statistical evidence for the genetics of chronic pain and the genetics of ME coinciding, because we suppose because of the same genetics underlying it. But interestingly, not all pain signals are replicated in our ME genetics. So here's not a genetic signal from ME at all. This is from a pain intensity genetic study that was published yesterday, and it indicates a gene called SLC45A4 as being directly involved in mediating the intensity of pain. And so this is the beauty of going genome wide, because then we can look in our Decode ME data and see here nothing. We do not see what was seen in this other study of pain intensity. And so for now, we have to ask ourselves about statistical power, etc. But for now, we can begin to say that the pain signals that that others have seen here do not relate to ME. And that allows us to hone the types of experiments that can be done, and to be more refined in what we mean by pain, because pain is very different for different people, and there are different types of pain. When the genes that underlie these genetic signals are looked at as to where they are most active in the body, there are 13 tissues that are most significantly active, and all of those are in the brain, thereby indicating the central nervous system as being the site of the ME genetic risk factors. So that's our main piece of evidence for saying that the neurological system is involved in ME. So what do these results mean for people with ME? We've received all of these questions, and they are absolutely the types of questions that need answers. Do the results mean anything for people with ME and their families? Well, the results from Decode ME, I believe, matter, but they matter on a population-wide average, and they do not say anything about individuals. And this is quite hard to explain, so I'll try and do so. And this is that each of the genetic risk factors are just slightly altering the likelihood that someone will have an ME diagnosis, only slightly. And that means that just because someone in your family might have ME, that does not mean that if they share genetics, that it increases the likelihood that someone else in that family will have ME by a large amount. It does not do that. And so, in essence, it does not have, our results do not have an implication for people's families. And that, in part, is because the genetic differences do not specify whether someone has ME or not. They are not sufficient to allow any diagnosis. And if you don't have any of the genetics that we have found, that doesn't mean say you don't have ME. Similarly, if you have all of the genetics, that doesn't mean to say that you do have ME. And that is because our study is all about population averages and not about individuals. And our study isn't about recovery, and it isn't about progression of the disease. It's about whether someone has a diagnosis or not. So Decode ME doesn't say anything about recovery, and it doesn't say anything about people getting better or not. We never said that Decode ME's results would immediately lead to treatment and diagnosis and testing. Of course, that's exactly what we want, but the genetics should have been done 10 to 15 years ago, and we should have had far better evidence for effective treatments and diagnosis now because of what was done, should have been done then. We don't have that, and so this is our attempt to kickstart a field that in the end will lead to effective treatments and diagnostic tests. Will our results have anything to do with other conditions such as fibromyalgia? I think so, yes. I think that there probably will be overlaps in these various conditions. And by knowing more about ME, we'll know more about fibromyalgia and the other way around too. And I think I've answered this question about whether a single participant of Decode ME will have any of these genetic differences, and the answer is not necessarily, because it isn't about individuals, it is about the population average. And we haven't been able to narrow down to single genes. And that is true across all of complex genetics. It's not due to Decode ME. Complex genetics doesn't tell us immediately what genes are definitely involved, but it does narrow down to a very small set of genes which we can annotate in particular ways to make some of higher priority than others. So here is our signal on chromosome 20, and you can see here is a very wide signal, and underneath them are all of these different genes, some of which we can determine some explanations for why people might have ME, because ZNFX1, for example, is an antiviral restriction factor, and we can make similar arguments for many of these other genes. But we do not know definitively which are the genes involved. But we can use the same trick that I tried to explain earlier. Previously, I talked about the coincidence of two genetic signals with respect to ME and chronic pain, but we can do the same thing with ME and the genetics of gene activity. So if the genetics of gene activity has a peak that doesn't overlay the ME one, then we can say that they are different. Whereas if they overlay one another, then we can say that there is some coincidence there which is consistent with that gene explaining the ME signal. So we've done that, and we've done that across 50 or so different tissues because genes' activities are different in each tissue. And the large circles here indicate which genes, so here's SUDS3, are active in each of these tissues, and their genetically determined activity coincides with the ME genetic signal. And the colors are different. You can see in green and in blue. The green one is where the increased risk goes with increased gene activity, whereas the blue ones are the increased risk go with decreased gene activity. And so that helps us to come up with hypotheses that if a gene is involved, is it because they are more active or less active with respect to ME? So what's next for us? We have still some way to go before completing all of the analyses. We want to do better gene prioritization, and we are making good headway with that. I've told you that we haven't reported on the X chromosome. We're about halfway through that. I didn't say anything about HLA, which is this region of chromosome 6, often to do with immunology. And that's because we haven't done the best analyses that we could do. And so we have to do that better. We haven't yet done replication testing using people with ME defined in the UK Biobank against their controls. We haven't used people from outside of Europe, their ancestries, but we are doing that, and that's called knockoff GWAS. We're using other algorithms, Targene and Gate, which have different benefits and drawbacks. And only when we've done all of those things to our satisfaction will we submit to a journal and then undergo the revisions and resubmission. But we hope to update our preprint before we submit to the journal. Now we have all of these 18,000 DNA samples that we have still in our freezer because we didn't use up all of the DNA for Decode ME, and that was on purpose because in Decode ME we're only studying the common DNA differences, but there's a different technology which is far more expensive called whole genome sequencing, which can look for rare variants and can gain insights into families that have rare variants. And we would love to do that. And we would love to do that whole genome sequencing with Decode ME samples. It's very costly. We're talking millions of pounds. But if we were to do this, we would find many of the genetic variation that are basically invisible, that are invisible to other approaches. Things that are, large chunks that have been duplicated or turned round, inverted or deleted, very sort of cryptic changes that are often not considered, but absolutely can be involved in disease. But to do this, we would need millions of pounds, and we don't have any funders for this project as yet. But something like £8.5 million would go a long way towards whole genome sequencing 10,000 samples. Finally, the data hopefully will have a long life, and people can ask us for the individual level data. Has to be done in a principled way, so we have to know that people are reputable, they're bona fide academic or commercial institution, that they have PPI in place, that they definitely will publish their results. But essentially, we want to fast track all of the applications and give them out to whomever needs them to answer these awful these questions, these difficult questions for this awful disease. So here are many of the people, not everyone, who contributed to Decode ME in the UK, and also want to acknowledge our international colleagues and collaborators who contributed the replication work. And hopefully I haven't taken up too much time, but hopefully there are now time for a question and answer session.
I turned on my camera and it doesn't seem to be just... My
apologies, I have taken up far too long.
No, that's... no, no problem. I'm just going to try to get myself visible again. I'm sorry.
I can see you, Trudy.
You can see me. Okay.
Yeah.
Just I can't see myself, but that's no big loss. I got a few questions, and I got a few questions of my own as well. I'll try to do some of the ones that have been written here first. There's someone who asks, she says that it has been discovered that ALS has genetic features in common with other neurological diseases like Parkinson, Huntington's, and dementia. Are there common genetic factors with ME as well?
Those signals that we have found are not in common with those other diseases that were mentioned, or in fact any other diseases that we could find. Where we have tested for the same genetics for two diseases, including ME, there was no evidence for that coincidence.
A bit in the same vein as well, that I know that people, among other, I think maybe Hornig has suggested that there's more than one type of ME based on symptom profiles. Like some people have more brain fog, some people have more pain, some people have more gut problems, and I think they even had something they called atypical, where people deteriorate quickly. Have you tried looking at your data based on these different symptoms profiles, or would you lose statistical power if you do so?
We will reduce statistical power. Will we lose it entirely? I don't know yet. But it is absolutely one of the things we would wish to do. And in part, we've done that by looking at people with infection versus no infection at onset, to think about how to separate people, to see if they're different. And in part, we've done that with the female-male analyses. But we'd like to do that in a data-driven way, to say, does the data separate people? And then if that happens, then are the genetics different between the, say, two different groups of people? And with colleagues, yes, that's underway too.
Okay. And then perhaps this is a stupid question, but there's something I'm trying to get my head around. I can understand the fact that you may have a genetic vulnerability, so that if you have certain of these signals, you're more vulnerable to get ME if you get an infection. So that there will be people who have these variants who do not have ME. But if there are people who do not have any of these signals and still have ME, would that suggest that what they have is a different disease? Or is it just that we haven't found something?
It is that. I do not pretend that the 8 signals that we have found represent all of the genetics. It's just the beginning. And in 10 years' time, I hope there will be other studies. And looking back from those studies, we'll see that Decode ME signals were just the starting point. And so if you don't have the genetic signals, the 8 of them, then you will probably have many others that have yet to be found. And there are also the strong contributions made by the environment and how the environment interacts with genes.
Okay. There is a question about autoimmune diseases, but you said you haven't looked at the HLA genes yet.
In the preprint, we say that there's a significant association in class 2 allele, but we had expected that association to go alongside 2 other alleles, and we did not see that. And we said that there was a better way of doing that analysis, which we will do. And so I will hold on and saying whether or not we have any evidence for an autoimmune component in the HLA until we do exactly the right analysis.
Okay. Because there was a Norwegian study some years ago that found an association with some HLA genes. Don't you have the data yet, or how does it tie in with the Norwegian study?
So I really do want to do the best study on our side first before we compare with anyone's. Otherwise, I may not be able to hold myself to the higher standards, or if we don't agree with the previous study, it may just be because we haven't done the right things in our study.
Okay. Then there's a question about how you included people, and how did you make sure that everyone satisfied the diagnostic criteria?
Yes, so we asked the questions that were relevant for the two international sets of criteria, the Canadian and the IOM criteria. And we asked people whether they had a clinical diagnosis of ME, CFS or ME, CFS. We asked questions about post-exertion malaise, so that if only when people answer those questions in a manner that was consistent with PEM, would they be included in the DNA portion. It will always be the case that someone will say, but you do not know that they have ME. Because what they have said is that they have a clinical diagnosis, etc. I think the study design that we have done, because we have found genetics, means that people absolutely have said what is true, that they have their diagnosis and that they have these symptoms. Otherwise, we wouldn't have seen anything, and we wouldn't have seen the things that we saw.
Okay. Do you think that these results also applies for children with ME? I would suspect so, since you have your genes from birth.
Yes, so our participants were 16 years of age and above. All of us are born with the genetics that we are studying. So if we had been studying the DNA of newborns, of those people who took part in Decode ME, we would have found exactly the same thing, because the genetics does not change.
Did you see any genetic association between ME and the depression or anxiety?
So because there are genetic signals for depression and anxiety in lots of places on the genome, there are close signals to the ME signals, but they do not seem to come from the same source. So they're close along the chromosome, but they do not trace the same paths, and therefore they do not seem to come from the same genetics thus far.
Yeah. Do you plan to try to get this study replicated in other countries?
Yes. We want our questionnaire to be used by anyone so that they can replicate. We want to ask the question of existing data, and that's in part what we have done in the preprint and will continue to do. I would hope that genome-wide association studies can be done in different populations. We are only studying Europeans at the moment. It needs to be done in diverse populations. But I expect there to be different signals in different populations, but also the same signals across different populations.
You talked about funding, and it has been notoriously difficult to get ME studies funded. We're now seeing lots of people coming down with long COVID, which looks extremely similar to ME in many cases. Do you think that this will make it easier to get funding for your next studies, or for this line of research at all?
I hope so. There are long COVID GWAS studies underway and also reported. We tried to gain funding for a long COVID study twice and failed. Absolutely, I hope that long COVID genetics and ME genetics go forward together side by side, helping one another.
So do I. We really need more research into this. Let me just see. Yes, I think I'm through the list of questions now. All I can say is thank you for an extremely interesting webinar. I think I'll have to watch it again later just to get all the details, but it was very informative. And
thank you so much for the invitation. And because essentially these are new findings, I am presenting them early, and so I won't have been as fluent as I would otherwise be, but I am very much appreciative of this opportunity to tell the story of the project and to tell everyone here as soon as we could have about the results, which I still think are incredibly exciting and hopefully groundbreaking.